Do mirror neurons have a role in goal-oriented reaching? Mirror neurons were first discovered in the 1990s. The initial study monitored electrical signals from specific neurons in area F5 of a monkey's premotor cortex. These neurons were monitored during grasping of different objects. As the researchers was handing the monkey an object, some of the F5 neurons started to fire. This surprising observation led the researchers to further investigate this finding. Their conclusion was that the neurons in the F5 that fire when doing an action will also fire when observing the same action. These neurons are called mirror neurons. Since this initial study, lots of research has been done on mirror neurons' potential role in movement execution. A basic understanding of human movement execution is necessary to analyze these studies. The motor cortex controls voluntary movement in humans. It sends axons that cross over or decussate, resulting in a bilateral system that controls the opposite side of the body in each half of the brain. Beginning in layer 5 of the motor cortex, around 400,000 axons go through the cortical spinal tract. Cortical spinal neurons connect to motor neurons and interneurons to coordinate muscle movements. The information they carry can be modulated in many other brain systems. In order to choose your movements, every part of the motor system must work together, including the motor cortex, thalamus, basal ganglia, midbrain, cerebellum, and spinal cord. Right next to the motor cortex are one of the areas containing mirror neurons, the premotor cortex. The premotor cortex controls movement and also responds to the movement of others. As previously stated, mirror neurons fire when a monkey carries out movements. When watching a human or another monkey carry out these movements, these neurons fire in the same way. These movements can be seen as a potential motor act. It's possible that this relates to the planning of motor actions. The movements and end goals of another monkey causes mirror neurons to fire in the same way because they are watching something occur, which as an outcome they are able to understand and predict. Motor imagery has also been demonstrated in humans in the premotor, parietal, and primary motor cortices. One needs to understand the goal of another's actions for them to fire. For example, hearing, chewing, or seeing half of a movement will still cause mirror neurons to fire, whereas a static image doesn't convey a goal, and does not cause significant mirror neuron firing. These inverse and forward models will be discussed within the context of a human reaching for a specific target, in this case a glass of water. Because movement is adjusted faster than humans can process sensory information, there is a reliance on proprioceptive information and an efference copy of the motor command. This example will focus on the biceps brachii and triceps brachii involvement. In reality, there are many other secondary muscles involved in stabilization and arm movement. The movement starts by using an inverse model. The inverse model uses proprioceptive information and sensory input to determine how to reach the glass of water. The primary motor cortex sends a signal from the representative area on the motor homunculus. As this signal is sent, an efference copy of the motor command is also sent through the thalamus to the cerebellum. This efferent motor neuron travels down the lateral cortical spinal pathway. This motor neuron travels through the internal capsule, cerebral pundicle of the midbrain, the pons, the pyramids and medulla, where the majority of the signal decussates. The signal continues down where it passes through the lateral column, the lateral intermediate zone, and then synapses in the cervical spine lateral motor nuclei of the ventral horn. From here, the signal synapses with alpha and gamma motor neurons that are sent to muscle fibers. The alpha motor neurons causes the extrafusal fibers of the triceps brachii to contract, causing elbow extension. At the same time, the gamma motor neurons fire, causing the intrafusal fibers to contract. This maintains stretch on the intrafusal fibers, which allows for continuous feedback through cation mechanoreceptors located centrally on the intrafusal fibers. This is called alpha-gamma coactivation. During this motion, the biceps brachii muscle spindle is lengthening. As this lengthening happens, the stretch receptors of the intrafusal fibers start to depolarize the sensory ends of the 1A and 2 afferent sensory fibers. The 1A fibers detect velocity of stretch and length, while the type 2 fibers just detect the length and position. the afferent sensory fibers, synapses at the dorsal root ganglion of the cervical spine. There are three important relays that happen in the dorsal root ganglion. One single signal synapses with an alpha and gamma motor neuron that is sent back to the biceps, causing an eccentric contraction. Another relay synapses with an interneuron that sends an inhibitory signal to the alpha and gamma motor neurons that stop the triceps from contracting. This process is known as reciprocal inhibition. This proprioceptive information is relayed up the dorsal column pathway, represented by the blue arrow. This signal travels through the caudinate fascicle up to the caudinate nucleus of the medulla. The signal synapses with another sensory neuron and decussates across the medulla, where it travels up through the medial lemniscus of the medulla, pons, and midbrain, eventually synapsing in the ventral posterior lateral nucleus of the thalamus. From here, the signal travels to Brodmann area 3A of the somatosensory cortex. A copy of this sensory inflow is also sent through the thalamus to the cerebellum. The other main proprioceptive response is initiated by the Golgi tendon organ. As the tricep brachii contract, the collagen fibers within the Golgi tendon organ increases in tension. This pinches the 1B afferent fibers, which opens mechanically gated stretch receptors, which cause an influx of cations, depolarizing the membrane of the sensory fiber. This signal propagates up to the dorsal root ganglion. At the dorsal root ganglion, the 1B afferent fiber synapses with an excitatory interneuron that sends a signal to the alpha and gamma motor neuron, which contracts the biceps brachii through alpha-gamma coactivation. There is also a synapse with an inhibitory interneuron that relays a signal to the alpha and gamma motor neurons that inhibits the contraction of the triceps. A copy of the sensory inflow is sent up the dorsal column pathway to the thalamus and then the cerebellum. This proprioceptive information from the muscle spindles and Golgi tendon organ is evaluated through a forward model by the cerebellum and predicts the endpoint of the movement. This information is compared with the efference copy of the motor command. If there is an error in this predicted forward model, an updated inverse model is created and is potentially sent through the rubrospinal tract. This sensory inflow and motor outflow is continuously assessed and used to correct the inverse model throughout the movement. This allows the movement to be continuous and smooth. This corrective mechanism happens until one reaches the endpoint, in this case the glass of water. Forward and inverse models use a combination of reflexive proprioceptive input, conscious sensory input, and unconscious efference copies to do a goal-oriented reaching movement. Some studies show that mirror neurons may be involved in inverse and forward model development. For example, this study focuses on the activation of mirror neurons when a monkey is carrying out an action themselves, without observing another. They found that mirror neuron populations fired before non-mirror neuron populations when executing an action. Modulation onset times were also earlier in mirror neurons, instead of being similar to non-mirror neurons during the action execution as previously thought. They actually have a head start. Observational visual information and the monitoring of one's own movements, therefore, may not be the only inputs and roles of the mirror neurons. This suggests that they serve to predict the end goal of actions and/or predict the sequence of movements needed to reach that end goal. Kilner and Lemon say that about half of mirror neurons are broadly congruent, while some discharge for specific action types. When it comes to humans, fMRI experiments have shown broad overlap between areas where monkeys possess mirror neurons and human cortical areas that fire when observing the actions of others, which makes the bold signals in humans consistent with the idea that we also have a mirror neuron system without outright confirming it. Single neuron activity from human neurosurgical patients has also contributed proof of mirror neuron activity in the medial frontal cortex and temporal lobe. However, the study did not look into the premotor or posterior parietal areas that we have focused so heavily on in monkey mirror neuron systems. Finally, they also go into the fact that firing rates for the motor neurons can be modulated by one's viewpoint, value, and in the absence of visual input. This means there is a top-down modulation via backwards connections from other neurons, with Jacob and Gene-Rod arguing that it is actually impossible for mirror neurons to function from visual input alone, as multiple goals could result in the same action. Predictive coding models of mirror neurons back this up, because through this model, mirror neurons are firing not strictly because of visual input, but because of a generative model predicting sensory input. This is similar to forward models and how the cerebellum analyzes an efference copy of a motor command when predicting expected movements. It is clear mirror neurons fire during movement and movement observation. Although predominantly studied in monkeys, there is some evidence for mirror neurons in humans. Mirror neurons preceding non-mirror neurons and functioning without visual input have similarities to forward and inverse models, although the exact role of mirror neurons in humans remains unknown.